What if you had a pair of scissors so tiny and so unbelievably precise they could actually edit the code of life itself? Well, that's not science fiction. Today, we're talking about the tool that made modern genetics possible, restriction enzymes. You can think of them as nature's very own molecular scissors. So let's just jump right into the problem. Imagine the entire human genome is a massive library, and inside that library are thousands of books, written with 3 billion letters. Now, your job is to find one single specific sentence in one of those books and cut it out perfectly without messing up a single letter around it. How would you even begin to do that? That was the huge challenge facing early geneticists. Because really, that's what DNA is, right? It's the source code of life. For the longest time, scientists could read it, but they couldn't really edit it. They just didn't have a tool that was precise enough to work on that tiny microscopic scale. I mean, how could you possibly build something that could find and snip one specific sequence out of billions? Well, here's the amazing part. The breakthrough didn't come from some brilliant human invention. It actually came from nature. The answer was hiding inside one of the simplest life forms on our planet, bacteria. And it was all because of a war that's been going on for billions of years. That's right. Bacteria are under constant attack from viruses, these things called bacteriophages. So, to survive, they had to evolve a defense system. They developed these special proteins, these enzymes, that could hunt down the DNA of an invading virus and just chop it into pieces. This effectively restricts the infection. And that, right there, is where the name restriction enzyme comes from. The journey from just seeing this happen to actually understanding it was like a classic scientific detective story. Back in the 50s, scientists just noticed this weird thing happening. It wasn't until the 60s that a scientist named Werner Arber said, hey, I think the bacteria are using some kind of molecular scissors. By the 70s, he, along with Hamilton Smith and Daniel Nathans, had proved it. And in 1978, they won the Nobel Prize for discovering the fundamental tools of genetic engineering. Okay, so how did these little molecular scissors actually get the job done? Let's pop open the user manual on this incredible piece of natural technology. So at its very core, a restriction enzyme is basically a protein with one super specific job. It zips along a DNA molecule, scanning it until it finds the exact sequence of letters it was designed to recognize. We call that its recognition site. And only when it finds that perfect match does it make the cut. Now, to really get how these enzymes are so precise, we have to talk about a simple idea you probably learned as a kid, a palindrome. A word like rotator or level that reads the same forwards and backwards. It seems pretty basic, but this exact idea is the secret to their accuracy. But here's the clever twist. DNA is double-stranded, right? So a DNA palindrome works across both strands. It has something called dyad symmetry. Take a look at the spot recognized by the famous enzyme EcoRI. The top strand reads GAATTC. Now look at the bottom strand and read it in the other direction. It's also GAATTC. It's a perfect mirror image. And that's the bullseye the enzyme is looking for. And scientists even came up with this really logical system for naming all these enzymes. Let's stick with our example, EcoRI. The E stands for the genus, Escherichia. The Co is for the species, coli. The R tells you the specific strain it came from. And that Roman numeral, the I, that means it was the very first restriction enzyme found in that strain. It's just a beautiful, clean system. But here's where it gets even more interesting. It turns out there isn't just one kind of molecular scissor. Nature actually created a whole family of them. And as scientists started discovering more and more, they realized pretty quickly that not all scissors are created equal. So scientists have sorted the thousands of restriction enzymes we know about into 4 main groups or types. And they're grouped based on things like their structure, what they need to work, and most importantly, how and where they actually cut the DNA. Okay, this chart might look a little technical, but I want you to focus on just one row, the very last one, cleavage site. Look at type 1, type 3, and type 4. They cut randomly or far away from where they bind. They're not very precise. But now, look at type 2. It cuts at or near the recognition site. It's simple, it's efficient, and it cuts exactly where you expect it to. That one little difference changes absolutely everything. And because of that incredible precision, today, when a scientist reaches for a restriction enzyme in the lab, there's about a 98% chance that they're grabbing a type 2 enzyme. They just completely dominate the world of molecular biology. So here's the bottom line. Type 2 enzymes became the go-to tool because they are predictable. The other types, they're like lining up a pair of scissors on a word, but then having them cut a random spot halfway down the page. Useless for precise work. But type 2 enzymes, they cut exactly where you tell them to. And that precision is what made them the perfect tool for genetic engineering. It's this incredible reliability that has earned them the nickname the workhorses of molecular biology. Seriously, they're the indispensable tool that you'll find in pretty much every genetics lab on the entire planet. All right, let's zoom all the way back out. We found this super precise, super reliable tool inside a tiny bacterium. What did that actually let us do? Well, it's really not an exaggeration to say it laid the entire foundation for the modern biotech industry. I mean, suddenly having the ability to reliably cut and paste DNA just kicked off a total revolution. It launched the whole field of what we call recombinant DNA, the art of mixing and matching genes. This single discovery led directly to things like gene cloning, DNA fingerprinting, and genetic testing. It even let us turn bacteria into tiny factories to produce life-saving medicines like human insulin. All of that started here. And so, what started as a simple defense mechanism, created in an ancient evolutionary war between bacteria and viruses, ended up giving humanity an almost godlike power, the ability to read, to edit, and to begin to rewrite the source code of life itself. You see, this discovery wasn't the end of the story, it was just the beginning. It blew a door wide open, and all kinds of newer technologies, like CRISPR, have walked right through it. But it all started with these little bacterial enzymes. And that leaves us with a pretty profound question. Now that we have the power to write in the book of life, what story should we write next?